everyone. In this chapter, we will talk about the forensic DNA analysis. DNA technology in forensic science has generated considerable excitement. In 1980, Wellman found a polymeric DNA locus characterized by a number of variable length restriction fragments called the restriction fragment length polymerism. Now, DNA analysis has been one of the daily work in forensic medicine. So, in the following chapter, we will talk about the forensic DNA analysis. In this section, there are three parts of DNA, that is, DNA markers, the DNA polymorphism, and the nomenclature of STR. The DNA structure is a double helix with the complementary strand linked by hydrogen bond. The basic unit of DNA molecular is the nucleotide, consists of 4 different bases, including the adenine, guanine, thymine, and the cytosine, with the deoxyribose sugar and the phosphate group. Nucleotides are linked by DNA polymerism to form a polynucleotide chain connected by the hydrogen bond between the adenine and the thymine, or the cytosine and the guanine. Within human cells, DNA is found in chromosomes. The human genome contained in every cell consists of 22 matched pairs of autosomal chromosomes and 2 sex-determined chromosomes. Thus, normal human cells contain 46 different chromosomes, or 23 pairs of chromosomes. Chromosomes 1 to 22 are numbered according to their relative size and occur in a single copy pair within a cell of nucleus, with one copy being inherited from the mother and the other copy coming from the father. Sex chromosomes are either X, Y for male or double X for female. Within human cells, there is a small circular genome known as the mitochondrial DNA. Mitochondrial DNA is inherited only from the mother and is located in the mitochondria with hundreds of copies per cell. When we talk about the DNA markers, there are some basic concepts we have to know. The chromosome location of a DNA marker is commonly referred as a locus. Through the Human Genome Project, thousands of loci has been characterized and mapped to particular region of human chromosomes. The alternative possibility of DNA fragment at a locus is termed as the alleles. A genotype is a characterization of the alleles present at a locus. Okay, here we use a capital or lowercase form to mark 2 different alleles at locus. For example, the capital A and the lowercase a. Then there are 3 possibilities of the genotypes: the double capital A, the capital A plus the lowercase a, and the double lowercase a. The double capital A and the double lowercase a genotypes are homozygous, while the capital and the lowercase a genotype, we call it heterozygous. Everyone is singular in the world, so is your DNA. The difference of DNA is termed as the DNA polymorphism. The genetic polymorphism within a population is defined as the presence of mutation at a frequency about 1% for a gene that is derived from at least 2 alleles at a given chromosome locus. This definition for genes could apply to DNA if the concept of alleles variation is taken as DNA polymorphism. DNA polymorphism is exhibited in the form of different alleles at a locus. There are 2 forms of DNA polymorphism, that is, the sequence polymorphism and the length polymorphism. The sequence polymorphism indicates the different single base, so we call it the single nucleotide polymorphism. And the length polymorphism are divided as the STR and the VNTR. The STR are the most commonly used markers now. DNA region with the repeat unit that are 2 to 6 BP in length are called the short tandem repeat, that is, the STR. These repeat units are arranged next to each other over and over again, look like just the serial batteries. The number of repeats represent different alleles at a locus. They are steadily inherited, but the number of the repeats in STR markers can be highly variable among the individuals. That's why the STR is effective for human identification purpose. STR repeat sequences are named by the length of the repeat unit. So, dinucleotides have 2 nucleotides in the repeat unit. There are also 3 nucleotides, tetranucleotides, and pentanucleotide. In forensic STR analysis, the tetranucleotide repeats are the most popular STR for human identification. Now let's see how the STR is named. The nomenclature for the STR is fairly straightforward. If a STR is part of the genes or fall within a gene, the gene name is used in the designation. For example, the STR marker TH01 is from the human tyrosine hydrolase gene located on chromosome 11. The O1 portion of TH01 comes from the fact that the repeat region is located within the intron number 1 of the tyrosine hydrolase gene. STR markers that fall outside of the gene region may be designated by their chromosome position. For example, the D7S820, the D stands for DNA, the 7 refers to the chromosome 7, and the S refers to the fact that the DNA marker is a single copy sequence. The final number 820 indicates the order in which the marker was discovered and the categories for a particular chromosome. Sequential numbers are used to give the uniqueness to each identified DNA markers. So, these markers means the 820th locus that has the single copies described on chromosome 7. Clear? In general, an allele at STR locus is labeled according to the number of the repeat unit. However, not all alleles for STR locus contain complete repeat unit. Microvariants are alleles that contain incomplete repeat unit. The most common example of the macrovariant is the allele 9.3 at the TH01 locus, which contains 9 tetranucleotides, repeats, and 1 incomplete. So for the incomplete repeat unit, we present it as the number of complete repeat unit plus decimal point and plus the number of bases. At last, let's see a picture of the position of the common used STR on human chromosome. We can see these DNA markers that are widely distributed on the chromosome include the X and the Y chromosome. Okay, in this section, we have talked about some basic knowledge about the DNA markers, especially the structure, the characters, and the nomenclature of the STR. So in the next part, I will introduce some techniques and methods for the genetic marker analysis. Thanks for watching.